Thanks very much. Is this one on? Pleasure to be here this evening. I'm actually a native New Jerseyan. Spent an awful lot of time in Chicago, and I don't get back as often as I'd like to, but it's nice to be back in this area. I'm going to talk tonight about what I think is potentially an enormously compelling public health problem, and that is the possible adverse health effects of cell phones and the radio frequency fields that are generated by those phones. The context of this is very simple. I'm sure everybody in the room has one of these in their pocket. There are currently more than 6 billion active cell phone accounts around the world right now. And it's estimated that sometime next calendar year, the number of active cell phone accounts will exceed the number of people in the world. So the human exposure is truly massive. The question is, does that exposure come with any toxicologic problems? Does it cause cancer? Does it cause other adverse health effects? So we're going to talk about this and what we really know and what this is going, how I'm going to frame this is actually in the context of a broader question in toxicology. Toxicologists are regularly called upon to draw conclusions based on incomplete data sets or perhaps conflicting data. And there are a number of ways that this can be done. What is most commonly done is with a weight of the evidence approach. So if you'd like to have a different topic for my talk, one that's perhaps slightly more academically oriented, this will be a weight of the evidence evaluation of the possible risks of exposure to radio frequency fields. I'm going to start with just 2 slides about physics. I'm not a physicist, I'm not an engineer, I'm a biologist, but it's important to understand what we're talking about here in terms of the waves to which we're being exposed. What we have here is the entire electromagnetic spectrum. Over in the left side, you see ionizing radiation, gamma rays, X-rays, and here's ultraviolet radiation, the visible spectrum. The area that we're interested in tonight is right in this area, in the low gigahertz range, about 2 gigahertz. And just as if from a historical context, I'll mention that the types of magnetic fields that are generated by things such as power lines, fluorescent lights, other electrical equipment are way down in here in the 50 and 60 hertz range. So again, I mentioned this to provide a little bit of context. We are looking at RF fields. We're going to be discussing those that are very close to microwaves or in the low microwave range, as opposed to these longer radio waves. It's important to note that these fields have dramatically different energies. Again, if you look at the gammas and the X-rays, we're talking about energies in the millions and thousands of electron volts. When you get down to the area 2 to 3 gigahertz, which we're going to be talking about tonight, we're down in the micro electron volts. So a difference of between 10 to the 6th and 10 to the 9th, between a millionth and a billionth in energy. Largely why this is non-ionizing radiation. And then the ELF fields, extremely low frequency fields, like come out of power frequency devices, have very, very small, down in the pico-electrovolt levels. So the basic question we're going to talk about tonight is, does exposure to RF radiation emitted by cell phones or other wireless devices increase the risk of adverse health outcomes? And I'm a cancer biologist by training. We're going to focus about cancer. That's the endpoint people are most concerned about. But there are certainly other possibilities as well. The analytical approach, as I suggested before, I believe this can be framed as a broader question in environmental toxicology, and that integration of toxicologic data into this weight of the evidence assessment that I talked about a moment ago is the best means to answer the question. I do want to talk about some very important variables to think about, especially if you have concerns about exposure to RF fields and possible health effects, how you might be able to limit your exposure. Clearly, dose-response relationships are critical components of any toxicologic effect. And within limits, if you increase the dose, you are likely to increase the response, increase the adverse effect. What impacts the dose-response relationships? Well, the exposure intensity. How much are you actually being exposed to? And this is a factor of a couple of different parameters. One is the amount of RF fields that are generated at the source. The second one is the distance from the source. And this is a very important determinant here. For example, the easiest way to decrease your exposure to RF fields emitted by a cell phone is use a Bluetooth device. If your concern is the amount of energy being deposited in the brain adjacent to your ear, the use of a Bluetooth device will decrease that energy by a factor of about 1,000. Electromagnetic fields fall off with the square of distance. So as the distance from the source increases, the field intensity decreases very rapidly. Whoops, sorry. The second, obviously, is the duration of exposure. And then third, and a very critical variable here, is a thermal effect versus non-thermal effects. One of the challenges with dealing with possible health effects of RF fields is that the device manufacturers say, well, our device, be it a phone or whatever, meets all FCC requirements. In terms of potential long-term health effects, that's a completely meaningless statement. Why? Because the FCC regulations were driven by thermal effects, by tissue heating. We know that any long-term effects of magnetic fields are going to result from effects that occur at non-thermal temperatures, where there is not obvious tissue heating. So the fact that you are below a thermal level says nothing about whether or not there is any risk associated with that exposure. Clearly, there are decades of research that say that thermal effects of microwaves, or by extension of radio frequency fields, can have adverse effects. They induce cataracts in the eye. Through tissue heating, they also negatively impact some temperature-dependent processes. And the most obvious example of this is spermatogenesis. But our concern here, again, is not with thermal effects, but is with possible effects that occur at much lower exposure levels. So I said this is going to be a weight of the evidence approach. What sorts of data can we evaluate? And there are basically three types of relevant data sets. Epidemiology studies, studies in human populations, studies in experimental animals, and then mechanistic studies, which don't identify something necessarily as being a hazard, but provide supporting evidence that something could have a negative health effect. So let's start with epidemiology data. People tend to look at that first because certainly if you demonstrate a clear adverse effect in a human population, you don't need to go any further. The story is over. So let's talk, though, about the strengths and limitations of different types of data sets. And something I tell my graduate students all the time, and although epidemiologists don't want to hear it, their data sets are not perfect. There are challenges there, too. There are some obvious strengths of epidemiology, however. First, as I said a moment ago, the studies are in human populations. You don't have to extrapolate from a rat or a mouse or a cell culture or from some other experimental system. If you see the effect in humans, you know it's relevant a priori. Secondly, the exposure levels are real world. People were actually exposed to the radio frequency fields at the doses that are being studied. There's no requirement to extrapolate from high doses to low doses as there is in experimental animal bioassays. But there are some important limitations, and sorry if I offend any epidemiologists in the group, and some of my best friends are epidemiologists. But you have to know both the strength and the weaknesses of your data set. Dosimetry and exposure metrics in this field are perhaps the largest single challenge. What were you exposed to? Oftentimes, the measure is crude at best. What's happened more recently is that epidemiologists studying human use of cell phones refer to the service company records, that the cell phone was engaged for X number of minutes a month for over the past Y number of years. And that's fine, but it doesn't consider the possibility that the individual was, for example, using a Bluetooth device. So we don't know what the actual exposure was at the target tissue. So dosimetry is a major challenge. Certainly, there can be confounding by unknown variables that you simply might not be aware could impact the endpoint you're looking for. And then finally, there could be selection or recall bias. And if anyone's interested, I can certainly talk more about these a little bit later on. So, does exposure to RF fields from wireless telephones increase the risk of brain cancer? This is the tissue of primary interest because of the local deposition of energy. What does the epidemiology tell us? Well, I'm going to show you a number of plots here. These are called forest plots, and they all have the same format. You'll see one here that'll have a line going up from it. This one means the risk in any exposed group is exactly the same as in a control group. There's no increase or decrease in risk. What you have in each individual study is a dot, which is a mean, and then the 95% confidence intervals for that observation. And as a general rule, if that confidence interval overlaps 1, this difference is not statistically significant. By contrast, if the confidence interval does not overlap 1, this is a statistically significant difference in that particular study. And then finally, there's a meta-analysis, which in this case shows that the mean of all these studies is almost exactly at 1. And I should say that in the interest of full disclosure, these forest plots were taken from a recent paper that was written by Mike Repiccioli, who's at the University of Rome right now. But I was a co-author on that paper, so I guess I'm allowed to steal the data. So what we can see is the risk of glioma, a particular type of brain tumor. If you ever used a cell phone, most of the studies demonstrate no statistically significant differences. 2 studies of note, one by Leonard Hardell from Sweden, showed a statistically significant increase. Another one, which is the largest epidemiology study ever done, called the Interphone study, was done by a consortium of primarily European groups and published in 2010. They show a small decrease. The meta-analysis put all of these together, no effect. There's no evidence that glioma risk is increased by every use of a cell phone. This is short-term use, and you can see basically the data are the same. An increase reported by Hardell, a decrease reported in the Interphone study. When you sum the data together, right on top of 1, no effect. And then longer-term use, again, same scenario. But overall, the sum of the data say there's no effect on glioma risk. Different type of brain tumor, meningioma. Ever used? Again, there was a small but statistically significant decrease in the Interphone study. None of the others were statistically significant difference. A slight decrease, but not significantly different when you put all of them together. Short-term use, no statistically significant differences. And then long-term use, and again, the number of studies is small. You have a statistically significant increase in the Hardell study, no significant effect in the overall. A third tumor type is acoustic neuroma. This is not actually a brain tumor, but it's a tumor of the acoustic nerve. And this is obviously a nervous tissue which is in the area of maximum energy deposition. And you see very similar patterns. Again, the only positives were reported by Hardell. None of the other differences were statistically significant. Most of the means were in fact below 1, and the meta-analysis shows no effect. Short-term use, essentially the same data. Long-term use, nothing statistically significant, and again, the meta-analysis, while suggesting a possible small increase in risk, was also not statistically significant. So what can we say in summary? Since 2000, more than 20 studies have been published in the peer-reviewed literature looking at the effects of RF exposure on incidence of cancer in various tissues. The vast majority of these have looked at brain cancer or acoustic neuroma or both. In the vast majority of these studies, no association was found between wireless phone use and cancer. Interestingly, only positive findings that have been reported all came out of Hardell's group. And it's interesting that Hardell is a Swede, and the Scandinavians have the best tumor registries in the world. So they know exactly how many tumors of different sites have been reported, have been diagnosed in the entire country. If, in fact, Hardell's data were accurate, one would have expected an explosion in brain cancers in Sweden over the last 10 years. That, in fact, has not been seen. So I think that leads us perhaps to question the veracity of those data. An interesting signal, however, in the Interphone study, although the overall trend was not statistically significant, they did report a small increase risk of brain cancer in the decile, in the 10th of the population with the highest use. Again, not statistically significant, not definitive, but this could be construed as a positive signal. So, what can we say in conclusions about the epidemiology? Well, the consensus opinion is there's no compelling body of evidence that says that exposure to cell phone radio frequency fields increase cancer in any site. But there are some very important questions that remain unanswered. First, is the risk of cancer or other health effects increased with very long-term use of these phones? Again, the vast majority of the population use of a cell phone has occurred over the last 10 to 15 years. What happens with tumors that have a 20 or 25-year latency period? Again, are the study designs sufficient for the study of cancers with very long latency periods? And perhaps most importantly, is the risk increased in sensitive subpopulations? And you can identify a number of sensitive subpopulations: people with a genetic predisposition to malignancy, people with exposure to chemicals or other agents that might increase the risk of malignancy, and perhaps most importantly, children. And we simply don't know whether they're more sensitive or not. So let's talk about some of the limitations to epidemiology and what you need to consider if you're evaluating epidemiology data. First, exposure assessment. And can people really, or can it be determined what people are exposed to? This is an ongoing challenge in the bioelectromagnetics arena. There's also a challenge with epidemiology in the limited sensitivity to detect a small effect on a finite but perhaps fairly substantial background. For example, brain cancers are relatively uncommon. You could identify a small increase in brain tumors. However, if the same effect was being seen in colon cancer or breast cancer or lung cancer, because of the higher incidences of those malignancies in the general population, we'd never be able to detect them. It's a statistical problem. And then finally, an issue of temporal variability of exposures. Do we know if there is a quote-unquote critical period when the organism is maximally sensitive? We simply don't know. And this adds some question to the epidemiology studies. There are some other limitations to epidemiology, though, that I think are specifically germane to studies of radio frequency fields. First, what's the relevant metric? And the challenge here is that cell phone frequencies have changed substantially over the past 10 years. 10, 15, 20 years ago, the signals were analog. Now they're digital. There are different modulations used. Can we lump these all together? Or are there specific differences in exposure that are important? We simply don't know. Secondly, again, the RF exposure in most epidemiology studies has generally been only for a few years or perhaps up to a decade. And we talked about the differential sensitivity of children and other subpopulations. So some Although the data to date, I think you can say, are pretty much negative, there are some positive signals, some questions remain. Perhaps the most important single limitation to a toxicologic assessment based on epidemiology is the latency of hazard development and the time required for hazard identification. What do I mean by this? Well, as I said a few minutes ago, there are currently 6 billion cell phones in use around the world. Let's operate under the hypothesis that in fact there is an increased risk of brain cancer associated with cell phone use. But that risk is very small, and it takes 25 years to develop. At 6 billion cell phones, if our risk was 0.01%, you would still have 6 million cases around the world. And they wouldn't be identified until people had been exposed for 20 years. Alternate strategies are clearly required for a potential public health issue of this magnitude. Where epidemiology data are inadequate or inconclusive, experimental model studies with emphasis on studies in predictive experimental models may provide critical data to identify and evaluate human health hazards. And I would propose to you that given the massive population exposure to cell phone RF, that this is an ideal example of a situation where experimental studies can provide critical information. In the interest of full disclosure, I'm an experimental biologist, so I'm preaching to myself at this point in time, but I hope to be able to convince you that these data sets provide a very important means to evaluate possible overall risk. Let's talk about experimental data. I'm going to start by results from 2-year cancer bioassays in rodents. These are considered to be the gold standard assays for cancer assessment. The strengths of the rodent bioassay is you know exactly what the animals are exposed to, what the field intensities are, for how long, by what schedule. These studies use models that are known predictors of human responses. There have been extensive evaluations performed by the United States National Toxicology Program and the International Agency for Research on Cancer, which is part of the World Health Organization, that have shown a very, very high correlation between positive results in 2-year rodent bioassays and positive carcinogenicity in humans. There's a lack of confounding you can control for virtually every possible variable in these studies. But again, there are some important limitations. Rats and mice aren't humans. We think that the biochemical and molecular systems are certainly comparable, but they're not identical. And there could be differences that arise as a result of these differences. Another factor is one from high dose to low dose extrapolation. In the particular case of RF, I'm not sure if this is a critical limitation because we are in fact limited by thermal stress. In many chemical studies, however, the high doses that are administered in bioassays may be 100 to 1,000-fold greater than humans would be exposed to, and the possibility of overwhelming a homeostatic regulatory mechanism that wouldn't happen at lower doses is always a possibility. But I don't think that's a critical limitation here. So what do the results of the 2-year studies say? And I'm going to go through these very quickly. The first one saw an increased total number of malignancies in animals exposed to pulsed microwaves. This is actually a very unusual endpoint. It's not generally accepted, but it's been published, and I thought it was, in the interest of full disclosure, I'd simply mention it. There was no increase in cancer incidence in any organ site in this study. This is the endpoint that is ordinarily used. But I'm going to present these 2 data sets for the other studies that were done. So you could say, depending on whether you believe this is a reasonable endpoint, this was either a negative study, as most toxicologists would believe, or maybe a weak positive. Another study done a few years later at Washington University in St. Louis saw no increase in the total number of malignancies and no increase in cancer incidence in any site. This was a completely negative study. Study done at Patel Pacific Northwest in Washington State. This was the Iridium Signal. You may remember some years ago, this was a Motorola product that was later discontinued, mostly because the phone looked like a large brick with a hot dog stuck on the top of it. It was too big and too heavy. Nobody wanted to use it. But the Iridium Signal was tested for carcinogenicity in rats. Again, a negative study. 2 studies done by a German group at the Fraunhofer Institute, again, one in Wistar rats, looking at 2 different modulations of cell phone frequencies, a completely negative study. The same group with a different lead investigator looking in mice actually saw statistically significant decreases in the total number of tumors and a decrease in the incidence of benign liver tumors. So clearly, if you look at all of these data together, there is no compelling evidence that these cell phone RF fields are carcinogenic in these bioassays. Co-carcinogenesis or tumor promotion bioassays are a little bit different, in which animals are exposed to the radiofrequency field and they're also exposed to some other agent that induces the tumor. And the question is, perhaps the RF field can't induce the tumor by itself, but it could enhance the activity of another carcinogenic agent. Strengths of these studies, again, you may be able to identify weak effects. You could also potentially identify effects that are only seen in sensitive subpopulations. And in fact, some of these studies have been done in genetically engineered animals to look exactly for that. Important limitations, though, these models have a very limited history of use in hazard identification, and we really don't understand how valuable they are in predicting human responses. So we have data sets, but they have a different set of positives and a different set of limitations than we talked about before. Obviously, the site of most interest is tumors of the central nervous system, and there is not a very good model, but it's the only model that's been used with some frequency in these studies with a transplacental dose of a compound called ethyl nitrosourea. Two separate studies done by Ross 80 at Loma Linda were both negative. Follow-on studies by 2 different investigators, Zook and Simmons, looking at a similar but not identical metric, also were negative. In lymphoma studies in a transgenic mouse that develops these preferentially, Mike Repiccioli, my colleague who I talked about when we were doing the meta-analysis of the epidemiology studies, when he was in Australia, published a paper demonstrated a statistically significant increase in lymphoma incidence in this transgenic model. We've used this model in our laboratory to look at cancer preventive agents, and it's pretty robust. I think it's a good model system. This obviously created quite a stir, and a number of laboratories felt it was important to replicate. In a second study, also done in Australia, using a very similar but not identical metric, but designed specifically as a replication study, failed to confirm this increase. And a third study done much more recently, again as a confirmatory evaluation, also failed to confirm the increase. So I think the consensus is that there was an issue perhaps in the exposures, and I'm not an engineer, but what has been identified is the fact that standing waves may have been set up and the animals may have been exposed to something other than to what was intended. Leukemia lymphoma in another model, in the AKR mouse, this is a very sensitive model to the induction of hematopoietic tumors by ionizing radiation. Negative study produced by Summer. A much more recent study by Lee in Korea using 2 different modulations, 2 different metrics, also saw no effect. And then mammary cancer, there have been a number of studies done, and again, all of these are negative. What are the issues? What are the challenges in these experimental bioassays? Not only the co-carcinogenesis or promotion bioassays, but the 2-year bioassays to date, one is group size and statistical power. Usually these studies have 20 or 30 or 40 or maybe 50 animals per group. So you have to have a fairly large effect to be able to identify it. I mentioned the fact that cell phone RF signals have evolved over time. What do we want to expose the animals to? And there are a number of experimental considerations. Were these signals generated and monitored appropriately? Do we know exactly what the animals were exposed to? How long were the animals exposed? Most of these studies, the exposure was limited to only 2 hours per day. And that was related to the exposure system, and the reason was the animals had to be restrained during exposure. These were what were originally called head-only exposures. So all the cell phone energy was deposited in the animal's head. In reality, they're head-mostly exposures, not head-only, but the animals were restrained so they could only be exposed for a couple of hours a day. And again, this possible sensitivity of subpopulations, and no one had ever studied young animals. So to address a lot of these issues, we're doing a study in our laboratory right now, supported by the National Toxicology Program of the National Institute of Environmental Sciences, looking to evaluate RF fields and address some of the limitations of studies that have been done in the past. And the principal participants, again, there are 3 of us in Chicago. Our primary engineers who designed and constructed the exposure system are from Zurich. And we are in about we've just finished the first year of our cancer studies. We've completed a couple of other studies at this point in time. The laboratory we build is very large. Vivarium is 17,000 square feet. It's fully dedicated to the project, and a lot of the details are not particularly germane here. Just to say that this is a very large operation. The laboratory itself cost about $9 million to build. We have 21 reverberation chambers, 14 rat chambers, and 7 mouse chambers. So we can expose up to 14 groups of 120 rats, or 7 groups of 224 mice simultaneously. So about 3,000 animals at one time. And each chamber is continuously monitored for the frequency, intensity of the RF signal, as well as for a variety of environmental conditions. Setting this lab up was no trivial exercise. This is our building in Chicago, and as you can see, we had to close the street. The laboratory actually starts in the back of the building, extends out underneath this plaza. We popped one of the roof panels off to deliver these through the roof. This is one of the chambers coming in and being moved into place. And then this is what, there are 7 of the chambers here, and there are a total of 21 in the laboratory. And to give you an idea why this was so doggone expensive, these are special RF doors that nothing will leak out. These doors cost $7,000 a piece. RF signals to which we're exposing Sprague-Dawley rats to 900 megahertz and B6C3F1 mice to 1900 megahertz. We're using different frequencies because of interspecies scaling issues, and we're using 2 different modulations. One is more of an American modulation, and one is used more commonly in the rest of the world. Importantly, our exposure systems were validated by the National Institute of Standards and Technology before we started, and they came back in before the start of the chronic studies. These are whole body studies, 10 minutes on, 10 minutes off, and the animals are exposed for 18 and a half hours a day. So the total amount of RF exposure these animals are getting is at least 10x what has been achievable in other studies. So we are confident that between the combination of greater exposure and much larger group sizes, if there is an adverse effect there, we're going to see it. The studies are fully compliant with good laboratory practice regulations. We first did a thermal pilot study, and what we were looking to do was identify RF flux densities that would not increase animals' temperatures. We implanted rats and mice with thermal microchips so we could collect the body temperature data, and our exposure groups were 4, 6, 8, 10, and 12 watts per kilogram. And again, the FCC limit is 1.6 watts per kilogram. So we are 6 to 8-fold above that. And we exposed animals of different ages, different sizes, and some pregnant dams as well for 5 days. And what we found in mice was essentially nothing. The mice tolerated the signals very well. But in rats, the highest field strengths, we actually saw mortality. We also saw body temperatures at specific absorbance ratios above 6 watts per kilogram. So as a result of this, the maximum exposure levels for non-thermal effects in mice could go higher than 12. In rats, we can only go up to 6 watts per kilogram. So these were the dose levels that were used for additional studies. We then went into pre-chronic studies, again looking for toxicity, pretty straightforward, using doses that were, or exposure levels that were based on what we saw in the thermal pilot study. We started these studies in the rats. The animals were exposed in utero. They started to be exposed at gestation day 6. So basically during the 2nd and 3rd trimesters through postnatal day 49, these animals were exposed. Standard toxicology observations. We did complete microscopic evaluation, all tissues, all animals, and the blood-brain barrier and the brain morphology evaluations are still in progress. And basically, we saw nothing. And this is actually good news. We saw no gross toxicity, no body temperature. The body temperature changes were consistent with what we'd seen before. No changes in the blood, no microscopic changes in tissues. And this is good for 2 reasons. Number 1 is that it confirms what others have reported. Number 2 is we know that people don't drop over dead and demonstrate acute toxicity as a result of using a cell phone. Had we seen that, there would have been a problem with our model. So we've now moved into the chronic studies. We want to see if these compounds are carcinogenic. Using 3 power levels plus the sham controls. Again, the rats were beginning exposures for pregnant animals at non-thermal levels. In mice, we're going not as high as we did in the perinatal study because we are concerned that we'd start to see premature mortality if we expose these animals for a long period of time. And again, these studies are going for 2 years. We have a standard series of post-mortem and in-life observations, the most important being microscopic pathology. The exposure completion in the mouse study will be next summer. The rats will be next November, and we expect to have the data out in the summer of 2015. So we have no final data to present there. I did mention mechanistic data, sort of the 3rd leg of the stool. And the presence of a clearly defined either physical mechanism, a mechanism by which this RF signal can interact with atoms, or a biological mechanism, alterations in a biochemical pathway, alterations in gene expression, would provide evidence supporting a possible hazard. Well, there really is no such data at this point in time. The consensus is that RF exposure is not genotoxic. It does not damage DNA. And there is no reproducible evidence of any mechanism at the whole organ or the whole organism level, at the organ level, cell, biochemical, or molecular level that is consistent with stimulation of tumorigenesis. So we're lacking a mechanism right now. The conclusion, there is no generally accepted mechanism by which RF fields may cause cancer. So what do we conclude here? Well, epidemiology, most of these studies have been negative. They found no relationship between RF exposure and cancer. The very few studies that have been positive have all been performed by a single investigator, and I think that gives us really some cause for concern. Certainly, we cannot ignore a positive signal like that. But I'd feel a lot more strongly that the signals were real if they'd been found by other investigators. 2-year bioassays are negative, and these assays have been demonstrated to be reliable predictors of cancer responses in humans, but the largest ones, the ones going on in our laboratories, aren't done yet. And I could change my tune a year from now. The multi-stage bioassays, most of these have been negative. There are a couple of them, there have been positive reports. None of them have been replicated. And they were all fairly unusual model systems that none of them had ever been used before for hazard identification. Mechanism of action, again, suggests no genotoxicity, and there's no other mechanism by which, a known mechanism by which RF may act to induce cancer. So the weight of the evidence evaluation, in my opinion, is the sum of the available evidence does not provide a compelling body of evidence to suggest that exposure to RF is carcinogenic in humans or, for that matter, in animals. There are positive signals there. Most of them haven't been confirmed. But there's some very important questions, most of which we've mentioned as we've gone through the discussion this evening. Is there a sensitive subpopulation who might respond to RF signals that would be fine if the general population was exposed to? Are there people with unusual sensitivities? We're not aware of any such subpopulation, but the possibility certainly exists. These people, as I said before, could have genetic alterations, could be exposed to other agents, it could be children. Does extended RF exposure induce adverse health effects? Exposure for periods longer than have been studied to date. And does RF exposure induce adverse health effects in humans? So what can I say in summary? Well, I think that probably the best and most objective study came out of the working group that was assembled by the International Agency for Research on Cancer. This group, again, IARC is part of the World Health Organization. This group met in June 2011, and the volume evaluating radiofrequency fields was published in last December or January. I was a member of this panel, and the conclusion was that RF fields fall into what is called Group 2B, possibly carcinogenic to humans. This was an extraordinarily high-profile event for IARC. They held a huge news conference at the end, which is very, very unusual. And this was covered by media all over the world. What sort of media coverage did we have? Well, this is one from CBS News, and I think it's really quite reasonable. Mobile phones may cause cancer, experts say. This, I think, was very reasonable approach. Another one from WebMD, expert panel, cell phones might cause brain cancer. Limited evidence suggests phones possibly carcinogenic. Again, this is accurate. Then there's one from Inc. Who links cancer to cell phones? Did the World Health Organization actually link cancer to cell phones? I don't think so. And you read the first sentence. A couple of days ago, the official party line was no adverse health effects. Not anymore. I think that's a bit of an overstatement. And then you go to another out of a British paper, Cancer Mobiles, World Health Chiefs in U-turn on phone fears. The media interpretation of the IARC evaluation was that cell phones are possibly carcinogenic to humans. I would propose to you that a more reasonable interpretation of this finding is possibly carcinogenic. Some positive signals are there that we can't ignore, but there's certainly no smoking gun. Thanks a lot. And if there are questions, I'd be happy to answer some. Well, if you're using it as a speakerphone, it's going to be several feet away from your head, so that the energy that's actually going to reach any structure in your body is going to fall off by 100 or 1,000-fold, something like that. So that if you use it as a speakerphone, if you use a Bluetooth device, you will decrease your exposure very dramatically. A question was also asked by one of the students this afternoon, what about now that most people, or a lot of people, text most of the time? I don't have any doubt that texting is going to decrease risk overall, because the risk occurs when you place the antenna in close proximity to a possibly sensitive structure. Your fingers are not going to be sensitive structures because hematopoiesis is not going on in the bones of your fingers. Overwhelmingly, yes. And I'll be honest with you, I was surprised that the Hardell studies were not kicked out. And in fact, there was an epidemiologist from the U.S. National Cancer Institute who was so furious that they were not kicked out, he resigned from the panel and left before the end. There were some weak positives in the animal studies, in the co-carcinogenicity studies, but the Hardell studies were certainly the driver why this was group 2B. And I should also point out that IARC, the WHO, has very, very rigid criteria for their compound classification. And basically, if the criteria, if you're in a zone, you have very, very little latitude in changing the classification. So, and they do that because the same criteria are applied to a broad range of chemical and physical agents. They're looking for reproducibility. A large fraction of us on the panel actually felt constrained by their criteria, but we had to play by their rules.
So when the cell phone is off, does it completely turn off all the waves, or are there very little?
If it's off, it's not doing it. It's not generating any signal. If it is on, but you're not speaking to it, there is intermittent communication with the closest cell tower. But the frequency and the amount of energy that's being emitted by the antenna is much smaller than it would be if you were talking with it. Way in the back?
I just read a paper about a recent study about cancers. They sequenced about 60,000 people with the cancer, their DNA sequence. They found the cancer generally take about 5 to 10 mutations to get, the cancer. The time period takes about over a 20-year period. So your study, I read mostly 2000 to 2008, and not over 10 years. So, and also I don't see any, sequencing anything. Do you see any SNPs, mutations happen in the sequences after your testing?
You're talking about the experimental studies? Okay, the experimental studies, we need to find an effect first. If we happen to see tumors, if there are brain tumors induced in these exposed animals, then the next step would certainly be to do some molecular analyses, and whether we're going in doing a series of southerns looking for mutations, or we're going to do microarray analysis and screen these tumors and look for differential patterns of cancer-associated genes. That's an obvious next step. Right now, I'm not sure what we would use as a substrate to test, because we simply don't have anything that has come out as positive yet. The 10 years, though, that's human data. You can induce cancer with some chemicals in rodents in as little as 6 weeks. And 2 years is considered to be a lifespan exposure in a rat or a mouse. That's why those durations are used.
Following question, and this may be a stupid experiment, but I'm figuring there's a lot of silly scientists out there. Has anybody taken some cells in culture and exposed them to some RF and see if there's some gene expression changes?
Oh, absolutely, absolutely. And there are reports. There's a journal called Bioelectromagnetics that's full of papers like that. The challenge is very often that one laboratory will be able to find it and other laboratories won't be able to reproduce it. The reproducibility is absolutely essential. My own bias is that these sorts of in vitro studies can perhaps direct you give you some ideas where you should be looking, but you can do all sorts of things in vitro that you can't do in a whole animal, and certainly are not going to happen in a human.
You mentioned that in your own lab study, the exposure to the rats or mice was something like 1 to 10 watts per kilogram. How many times is that compared to a regular exposure that a cell phone would?
Well, it depends on how you use your cell phone, but as I said, the FCC limit is 1.6. Okay, so you're about six-fold, but again, we're looking at exposure for 10 minutes on, 10 minutes off for 18 and a half hours a day. You tell me, my phone sits in my pocket and I probably talk on it 2 hours a month. What does a heavy cell phone user use? 4 hours a day? I know there are some people who, salesmen and stuff who are probably on their phones 4 hours a day. I honestly, I couldn't tell you what like the top decile is, what the median is. I would suspect that it's probably up in the several hours per day. The challenge is with people texting more and more that number's changing. It's a very fluid exposure scenario.
So I was just wondering if anyone looked into, there's a lot of phones, they double its data devices, and Wi-Fi frequencies can be 2.4 gigahertz, 5 gigahertz. So that's a little higher than the cellular frequencies, if anyone took a look at that, because you can have data coming in while you're on your phone.
The overwhelming amount of exposure you're going to get is when your phone's transmitting, not when it's receiving. But but it does point out a good question, and one that I mentioned briefly in passing is a 2 gigahertz signal different from a 2.1 gigahertz signal? One of the things that we're finding, actually, we're using two different modulations, and the GSM and a CDMA, which is actually an IS-95 modulation, and we're seeing slightly different effects on body weight, and the GSM has a little bit extra energy, more energy in there. But the challenge here is that these are very, very costly studies to do. The program that I talked about, the total budget is just over $25 million. We'd be happy to do it again if you'd like to write us a check, and in fact, we'll even put your name on the top. Because we're going to have a facility that's going to be ready to go in about 15 months. I'm not sure those questions are ever going to be answered very effectively.
So, Dave, the industry that produces cell phones, when they probably look at your collection of data from IIT, they've probably been pretty happy with what the results have shown. That is, no demonstrable effects. But then when they look at the IR scientific interpretation, they were probably pretty, maybe surprised, maybe not surprised, but they probably weren't happy with it. What do you see the industry's position in terms of, the whole area of possible carcinogenicity from cell phone exposure?
The industry's in a difficult position because they have no good way of actually getting a specific question answered that they want answered. Why? Because if Motorola comes to me and says, I would like you to do this study and we'll pay for it, well, then suddenly I've taken industrial money, and so I'm now a bad guy. I'm wearing the black scarf, and anything negative data I generate is an industrial fix. I don't believe that happens. I really don't. But they are looking to certainly try to raise questions about the validity of some of these studies. Some of them have been very critical about the Hardell work. And it's not only industry people, a lot of United States government and epidemiologists have been very, very critical about that work too. My guess is if I were working for them, I would just sit back and see what blew over. I can't say how they're going to do it, because again, if they try to fund their own studies, those are going to be looked at by certain segments of the population with a really jaundiced eye. They're in a no-win situation.
What would it take to actually reach a conclusive result? Like you mentioned, there's a lot of studies, and most of them have a negative result. So if 10 more studies are done and they all show a negative result, would you do another study? Like what would be the way to get a definitive answer and make that possibly be,
Well, and I think you have to break that question down into pieces, and it's a very important one. And I'll start with the animal studies, because that's probably the easiest place to start. If our animal studies come out negative, my guess is that there will never be another animal study done unless the cell phones change so much that the signal is substantially different than it is right now. These studies are just too expensive and they take too long. The only thing that I would see that would really trigger shall we say, a genesis of new research in the field were if a mechanistic scientist came and identified a pathway or pathways which was demonstrated to be causally linked to neoplasia in a specific target tissue, and then other people went out and independently confirmed that, replicated it over and over and over again. Then suddenly we've got a signal. That still doesn't mean that there's going to be an adverse health effect, but it would lead us in the right direction. My guess is the epidemiology studies are going to continue for another 25 years. And the challenge then, as they continue, we want to know if kids who started using a cell phone when they're 6 or 8 are more sensitive than everybody else. We don't know that right now. But as we've already discussed, the exposure is very fluid. It's a dynamic process, and not only are the signals changing to what people are exposed to, showing qualitative changes, but quantitative changes as well. And if people stop talking on the phone and text 100% of the time, the problem's going to largely go away.
Has there ever been a study where the frequencies were tested on an animal that actually had cancer and its effects on increased metastasis or
There have been a number of studies looking at RF exposure in animals that have cancer. And in fact, very, very high levels of RF have been studied as possible therapeutic approaches. Whether or not that's simply a function of local tissue heating, I think, is unclear at this point in time. There are a number of this could potentially be combined. Let's say if you were administering a drug in a liposome or something like that, and you had a very limited exposure at the tumor, you heat the tumor up, the liposome hits the tumor, the liposome releases the drug at the target site. Those studies have been done. I'm not aware of any large series of studies that's looked at metastases. Most of it is, if it's not been looking at a hazard identification, has been much more focused on the possible use as a therapeutic. In the back. Pardon me?
What seems to me to be the most convincing argument is that these RF fields have a much lower energy than even something like, say, visible light. So how do the scientists who believe that there is a possible carcinogenic effect explain this?
That's a great question. And first I'll say that if indeed these RF fields cause cancer, I don't know how it happens. But I will step back a moment and say some years ago, and this started off looking at extremely low frequency fields which have much, much lower energy still, some physicists, some well-known physicists said this cannot possibly happen because the field strength, the energy level is too low. One of the challenges with this area is that engineers and physicists need to talk to biologists. The biology or the physicist who made that statement was a very smart guy. Unfortunately, he ignored some very fundamental understanding of mammalian physiology. It's signal amplification by a specific receptor. And the best example of this is the amplification of visible light by the retina, where the energy that impinges upon the retina can be amplified as much as 10 to the 7th. A huge increase. So I would propose that if in fact there is a health effect or other biological effect of the RF exposures, that this happens through some receptor-mediated signal amplification mechanism or signal amplification cascade. The challenge is no receptor's been identified. We don't have any idea what the target would be. So I am not willing to completely discard the possibility of an effect based on the physical argument. I'm not nearly as confident as some physicists are that it absolutely cannot happen.
There have been studies that may suggest that cell community
You know, this is something that is most likely, and perhaps you can argue unfortunately, going to have to be done in humans, because these sorts of behavioral bioassays, humans are can be done much more sensibly in human populations. You'd have to have a big effect to see it in an experimental animal. I know some of those have been reported. Reproducing those, what I would call sort of soft endpoints, tends to be a challenge. But I'm sure that people are going to continue to look at those. And again, the definitive data is going to come out of human studies.
Is there any correlation between cell phones and laptops, since they're also wireless devices?
Laptops, wireless LANs, I mean, all of them use similar types of signals. The critical difference is, unless you're in the library and it's 11 o'clock and you've had a big dinner, you're not likely to put your head right next to your laptop. Maybe some, the proximity issue is a very important one. If your laptop is emitting signal, first, if it's going to a local wireless LAN that's 100 feet away, the total flux density that's going to be generated is going to be relatively small compared to a cell phone that's communicating with a tower that could be a half a mile away or 3/4 of a mile away, something like that. So that's one. And then number two, the fact that your head is 2 feet or 3 feet or 4 feet away from the laptop means that signal is going to fall off by another factor of 100 to 1,000.
So how about, like, I'm a notorious texter. I like to lay down in bed and text at night. So how much does the factor decrease when I'm sitting there at night and having the phone that close to my face and to my eyes, where the eyes are directly connected to your brain and are just more susceptible to light? Is that also like a risk? Is the risk the same as me holding the phone up to my ear?
I certainly wouldn't think so. I mean, again, you're talking about a distance of maybe a foot or 18 inches, something like that. The fields are going to fall off substantially through that distance. So again, the distance is a real driver. And again, this is a situation where if you're concerned about exposures, the Bluetooth headsets, especially if you have one, you're in your car, you put your phone in the console or something, you're getting virtually no exposure at all. I don't think that sort of texting is going to generate much exposure at all.
I hope this is an inappropriate question, but as a cancer researcher, is there something you might know about preventing cancer that we in the general public don't know?
Well, I would hope I know more than the general public does in a number of arenas. I'm not always sure about that, at least when I read the reviews of my grant applications. As I was telling a group of students this afternoon, my take-home message, and this is a completely different topic, but what I spend most of my life doing is trying to develop drugs for cancer prevention. I started off in breast cancer. We work now more in oral and prostate cancer, but work in some other tissues too. I think the overarching message is eat what your mother tells you. Vegetables and a high vegetable, high fiber diet. I mean, the diet. There's no question that your diet can have significant impact on modulatable cancer risk, because you can't pick who your parents were. Your genes may be the single most important single risk factor, but you can't change those. What you can modulate is the potential promotional or anti-promotional activity by doing things like not smoking, not drinking to excess, and eating a diet that can contain a lot of antioxidants and things like that. In terms of pharmaceuticals and things, the only compounds that I really like in terms of with their toxicity to efficacy ratio are low doses of some NSAIDs. Aspirin and ibuprofen and naproxen and a number of those compounds have cancer preventive activity in a number of different organ systems. And at low doses, their risk of cardiovascular events and gastrointestinal bleeding is very small. Yep. again, think about the distance. You know, if they're more than 2 or 3 feet away, the amount of energy that you're going to be absorbing from those cell phones is near zero.
I have a question regarding the duration of exposure. You defined the short term and the long term. Are you revisiting that definition with increased usage on human population?
You know, all I'm doing is repeating epidemiology data that's out in the literature. You know, these were not studies of my design, and I think, and the set points for short-term versus long-term, I think were less than or greater than 1 year, but I can't be certain about that.
Thank you so very much. We do need to wrap it up, but Dr. McCormick, I'll speak for him because I've known him a long time and I can speak for him. I can say that he'll be available up here for a few more moments if any of you are interested in talk to him a bit longer. But I do want to thank you for saying, listen to your mother. Now, not only because of food, but we do know best because we have gone through it and we've more or less experienced things a little differently and a little longer than you have. And he has said it twice, so I think he's saying that to my benefit. So thank you very much. Thank you all for coming. Our next speaker, if you enjoyed this one, is on May 1st, Dr. Eric Green from the National Institute of Health, and he is the National Director of the Human Genome Project. So we'll see you then. Thank you.